Hello everyone, I'm Vizueta and I'm going to talk about the Global and Genomic Alliance, the GAGA project, and I will give a brief update about the current status of the project and also the potential of our dataset and also the comparative genomic analysis in the ANT-taxa. So first of all, why the ants? Well, one of the key biological aspects of ants are their sociality. So they have an advanced division of labor in which they have morphologically distinct castes that they use for breeding. For example, the queens, for foraging and nursing, such as the workers, and some species have even soldiers for defense. So this has made ants like impressive social innovators, and we find across the ant tree of life many social complexities and also different adaptations, such as, for example, some of the species. So mutualism with plants, we have also the amazing farmers that are the ants from the genus Myrmicine that they cultivate their own fungus cultivars, and also some species that display these mass breeding columns or the foraging trails that you might have observed in nature. So the main goals of the GAGA project is to sequence one representative per genus, but not only get high quality genomes, but high resolution transcriptomes, micro-CT scans, as well as life history, microbiome surveys, and butcher materials. So GAGA started in 2017, and with a pandemic between the middle, we have been able to finish the first phase of sequencing, in which we have generated 163 genomes. So we have a pretty good coverage across the tree of life and also a good diversity of fund and different life history traits. So the main strategy for sequencing was to retrieve high quality PacBio long reads to combine with short reads for policing and, if available, sequencing high C. But the little biomass available of these tiny organisms make complex the sequencing, and some of the species we could also sequence with short reads, but most of them fall in the PacBio plus short read sequencing. And we were able to improve to 16 the number of chromosome-level assemblies in ant species. So that's a great number that we have now in ants to explore, for example, macrosynteny across the ants. With these high-quality genomes, we conducted gene annotation that you can see here. The number of genes varies between 11 to 14,000, and you can see here genome size that also varies across the ant phylogeny. Importantly, we conducted a gene reannotation of many gene and gene families of interest in ant evolution, such as, for example, the chemosensory receptors that are involved in the social communication, as well as immune-related gene, neuropeptides, and a list of 400 gene and gene families. So for this, we use mostly Bitacora, but some of these gene families were also curated in Apollo, such as this cluster of odoron receptors. And briefly, we conducted orthology assessment, and across 90% of the species, we identified 8,000 single copy orthologs that are conserved across our ant, 90% of our ant genomes. So using also this high quality genome, we conducted for the first time in ants a whole genome alignment, for which we use two methodologies. One is Cactus, that is a reference-free genome alignment, and multi-C based on monomorium Pharaonius reference. Both are pretty similar in terms of coverage, and here highlight that for 80% of the species, we found around 100 megabase aligned. So we can use this to explore like macro and micro synteny across ants, and here an example are the highly conserved elements using a fast cons analysis that we uncover. So we uncover 35 megabases of highly conserved elements that make up about 10% of the genome sequence across all ants, and we see an enrichment in coding regions as we would expect, but we also uncover some intergenic regions that might be important regulatory elements across the ant genomes. So we conducted the phylogenomic analysis, and I would like to highlight here that we did for the first time a real phylogenomic analysis using the whole genome alignments, from which we extracted intergenic regions and reconstructed the phylogeny for our species. We uncovered the backbone of the phylogeny is similar to that previously proposed in the literature, but we uncovered some new relationships across one of the subfamilies and also the placement of certain genus. Using this phylogenetic framework, we can explore now the genomic implications and genomic context of different life history and phenotypic traits. So we collected surveyed different social systems like traits and ecological traits across our species, as well as we have like morphological data from 76 of the species for which we have micro-CT scans. So with this all together, we are now conducting the comparative genomics analysis. And one of the most important and interesting questions in us is the genomic mechanism underpinning social complexity. So here you can see like the ancestral reconstruction and trait mapping of two traits that are related with social complexity, such as colony size, that is the logarithm of colony size, and worker polymorphism, whether the species have monomorphic or polymorphic workers. And what we are currently working right now is in underpinning the molecular mechanisms associated with this trait evolution. So we are conducting DNDS analysis and, for example, exploring positive selection related to convergent evolution based in this transition from worker polymorphism for colony size and these social complexity events, as well as expanded and contracted gene families that may be associated with these traits. In addition, we are also exploring gene family evolution, and one of the most important in this sense are the odorant receptors that you can see here. We found a wide number of odorant receptors across all of the Andes species, ranging from 200 to even 500 of receptors, and they are immensely expanded compared with other insects. And we do find a correlation with the transposable elements. So it has been hypothesized that the transposable elements contribute to the tandem duplication of odorant receptors, and we do find a correlation in our own genomes. Another sensory perception are also the visual systems. So we annotated the opsin gene families across the genomes, and here we have a much more conserved number of opsins across all the species, being most of them single copy. But we found here is that transcriptional regulation of the expression of these opsins, especially in those species for which that are hypogaic and they have absent eyes or reduced eyes, compared to those present of or developed eyes. So what we are seeing is the regulation in the expression of these opsin gene families in these species that have absent eyes. So finally, I would like to acknowledge all the people that has contributed to the project and belong to the GAGA consortium and the organizer for this event. So thanks to you for your attention.